We've been talking about the causes of evolution, but how do we know when evolution is actually happening? Well, if we can collect some sort of data, get some sort of numbers to explain what's happening in a population, then we can define when we think evolution is actually occurring. So we defined evolution as a change in allele frequencies over time. So if we can determine the frequencies of an allele in a particular population, and we can look at it at different stages in this population, then we should be able to determine whether or not that allele frequency is changing. So we can calculate allele frequencies and calculate how a population is changing by using the Hardy-Weinberg equation. So the Hardy-Weinberg equation is used to observe changes in allele frequencies and also to help us determine which factor is causing evolution to occur. Now, in order to use the Hardy-Weinberg equation, we need to make sure that our population is in Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium means that a population is not evolving. Essentially, it means that a population is stable. Now, populations are never stable. They're never not changing. So if we're going to use our Hardy-Weinberg equations, we have to assume that we have a hypothetical population or a hypothetical point in time where a population is not evolving. So in order to use the Hardy-Weinberg equation, we need to make some assumptions or set some conditions in order to use this. And that's simply because changes are occurring all the time. So we can assume that if our population was not changing, all of these conditions would be met. And so when we begin to look at the population, we can try to determine which one of those conditions is being violated and therefore which condition is causing our population to change. So in order to use the Hardy-Weinberg equation, we need to make a few assumptions. Basically, our assumptions are that there are no causes of evolution working on that population. So we are going to assume that there's no natural selection, that there are no mutations occurring in the gene that we're looking at, there's no migration or no gene flow, that our population is infinitely large, or that genetic drift is not occurring. So we're making the assumption that none of the mechanisms that we've talked about are occurring in that population. Now we also need to make one more assumption, and that assumption is that there is random mating. So what this means is that for the gene that we are looking at, for the allele frequencies that we're looking at, individuals are not choosing to mate with a particular phenotype or a particular allele. In other words, every allele has an equal chance of mating with every other allele. So if individuals tend to mate within a particular phenotype, it means that random mating is not occurring. So this last one, random mating or no inbreeding, this assumption actually doesn't cause evolution to occur, but it does cause genotype frequencies to fluctuate. So if individuals are choosing particular alleles, then those allele combinations are going to become more frequent. Now, over time, this could potentially lead to evolution happening, but random mating, non-random mating in itself is not really a mechanism of evolution, but it is something that we have to assume is happening if we're going to use our Hardy-Weinberg equation. So our Hardy-Weinberg equation is based on the total alleles in our population. So just by convention, we usually use the letter P to represent the frequency of the dominant allele and Q to represent the frequency of the recessive allele. Now, if you are calculating a frequency, you are going to take the number of items in a subset divided by the total. So for example, when you calculate a grade on an exam, you are doing a frequency that you're then converting into a percentage. So you're taking the number of points you earned divided by the total. That's actually a frequency. When you multiply it by 100, you get your percentage. So a percentage is a good way to think about what we are looking at here. You're looking at what proportion of the population has each type of allele. Now, the frequencies of each of those alleles is always going to be equal to 1. So if you're sharing a pie with a friend and you eat 3/4 of that pie, your friend gets the last remaining quarter. If they eat 2/3 of the pie, you get 1/3. So if one increases, the other must decrease because they're all part of a whole. Now, the actual Hardy-Weinberg equation is the equation written down on the bottom. Those P2 should actually be P squared, and I am 100% certain that I've fixed this about 7 times. It should say P squared plus 2PQ plus Q squared equals 1. Now we get this Hardy-Weinberg equation as simply the binomial expansion of the the allele frequencies. A binomial expansion is just when you multiply a two-factor equation by itself. So P plus Q times P plus Q. I don't expect you to do that, but it does help us understand where this equation comes from. P squared, the first part of the Hardy-Weinberg equation, is going to represent the frequency of the homozygous dominant genotype. So basically the chance of getting that P allele 2 times. So the frequency of getting it once times the frequency of getting it twice, because remember, all individuals get 2 alleles, one from mom, one from dad. Q squared over there on the right is the expected frequency of the recessive genotype. In other words, the chance of getting the recessive allele once times the chance of getting it from the other parent. In the center there, we have the heterozygous genotype. So the chance of getting the P allele and the Q allele. Now there's a 2 there, and I will explain to you why that is in just a minute. But the Hardy-Weinberg equation is actually looking at genotype frequencies. But those genotype frequencies are based on allele frequencies in the population. So remember from our unit on genetics, each individual is getting an allele from each parent. So this Hardy-Weinberg equation represents all the different combinations of those alleles in the population. So this image here shows how we get our P squared, 2PQ, and Q squared. So if we are looking at a population where the frequency of the red allele is 0.8 and the frequency of the white allele is 0.2, these are all the possible individuals that we can get. Now this example is actually an example of incomplete dominance, and that's where neither trait fully covers the other and you get an intermediate trait showing up. And the reason that our book does this is so you can see where all of the alleles are hiding. So P squared is equal to the chance of getting the red allele from one parent times the red allele from the other parent. 2PQ is the chance of getting one white allele from one parent and a red from the other. But that 2 comes from the fact that we can also get the combination the other way, the red allele from the opposite parent and the white allele from the opposite parent. Q squared comes from getting the white allele from one parent, the white allele from the other parent. So on the outside, we see our allele frequencies. On the inside, we see our genotype frequencies. Now, if you're looking at this closely, you're probably thinking that this is just a Punnett square, and it really is. And that's why the Hardy-Weinberg equation works, because it's based in inheritance. It's based in genetics. So the basis of this equation, though, is being able to calculate allele frequencies. In order to do that, we need to start with the total number of alleles in a population. So keep in mind that for every individual, they carry 2 alleles. Individuals that are homozygous, so the red flower or the white flower, have 2 identical copies of their allele. And an individual that is heterozygous, this pink flower, has 1 copy of each allele, but they all have 2 copies of any allele that's present. So let's look at our flower example. This is snapdragon flowers. And let's say that we have a wild population of 500 flowers. Now there are 2 alleles. We've already been talking about these, the red allele and the white allele. And let's say that in our population, we have 320 red flowers, 160 pink flowers, and 20 white flowers. So I would like you to pause and try to calculate the allele frequency for the red allele and the white allele, and then come back and see if you were correct. So in order to calculate our frequency, we need to know the total. So if there are 500 flowers and each flower has 2 alleles, we have 1,000 alleles total in our population. Let's start by calculating the frequency of the red allele. So for our red allele, we first need to know the total. We already calculated the total allele as 1,000. Now we need to know the total number of red alleles. So our individuals that are red flowers have 2 copies of the red allele. So within the red flowers, we have 320 red alleles and 320 more alleles. Now we also have some red alleles down here in these pink individuals. So the pink individuals have only 1 copy of the red allele. If there are 160 pink flowers, that means we have 160 pink alleles located in those flowers. So our total number of red alleles is going to be 320 plus 320, because each red individual has 2 alleles, plus the number of alleles that are found in those pink flowers, 160. So the frequency of the red alleles is going to be 800 divided by 1,000, or 0.8. Our frequencies are always going to be written as a decimal, not a percentage, unless I specifically ask you for the percentage in the population. Just go ahead and leave it as a decimal. If you need to round, round to 2 decimals, unless it says otherwise. So now let's calculate the frequency of the white alleles. So again, our total number of alleles is 1,000. So now we need to determine how many white alleles are present in the population. So keep in mind that each white flower has 2 white alleles. So in our white flowers, we have 20 white alleles plus 20 white alleles. And then in our pink flower, each individual has 1 white allele. So we're going to add in the 160 white alleles from the pink flowers for a total of 200 white alleles out of 1,000. Which is going to be 0.2. So our frequency of white alleles, this should say white here, not red, should say white. Our frequency of white alleles is going to be 0.2. So to move on to the Hardy-Weinberg equation, we can actually plug in our allele frequencies to determine how many individuals we expect for our homozygous dominant, or in this case our homozygous red, how many individuals or what proportion of the population we expect to be heterozygous, and what proportion of the population we expect to be homozygous recessive. Because these are frequencies, they're always going to add up to 1 because these allele combinations are going to make up the entire population. So we're going to do some practice problems in class. I would love for you to practice filling in this equation, but we're going to do some actual examples during our in-class time.